I'm delighted to welcome Professor Robin May.
That's quite the introduction. Thank you. Good evening. Welcome. Thanks for joining us either here or online. I'm Robin May. This is the second in the series of our discussions around immunity. And today, I want to think mostly about our own immune systems, how they work, why they sometimes go wrong, and what you can do about it. And I want to start by getting you to imagine yourselves back in 1882, okay, and particularly because it's a really grotty evening here in London, imagine yourselves on the much balmier shores of Sicily, okay, in the Mediterranean, with the sun beating down, resting gently on the beach. And in one hand, you have a bottle or a jar full of these things here, which are little starfish larvae. And in your other hand, you have a thorn that you have plucked from your tangerine tree outside your front door, and you can see where this is going to end. You're going to go inside from your wonderful beach, and you're going to set up your Victorian-era microscope. You're going to put your unfortunate larva under the microscope, and you're going to stick your thorn into it, because you are a scientist. And then, like all good scientists, you're going to go off and have a good meal, nice sleep, come back in the morning, look down your microscope, and what you're going to see is this. You're going to see that where you inserted that thorn, cells, all looking roughly the same, have gathered around the site of insertion, around the prick where you put into the larva, and they have gathered and they've got accumulated this debris in the middle. And you have just made the most fundamental discovery and the first notable experiment that we call immunology. Because you're Eli Michnikov. Michnikov was a Russian-Ukrainian scientist who had, and rather ironic given where we are today, had fled unrest in his native country, set up a lab in Sicily, and was interested in how animals generally respond to threats, to infectious threats or to foreign threats. And he used these marine larvae as his model. And in doing this, and he writes in his diary actually that he was too excited to sleep that night, but he just had this expectation that something amazing was going to happen the next morning. And he saw this and he recorded it. And he quite rightly said, I propose that there are specialized cells in animals whose job is to defend them against foreign infectious threats. And he was bang on the button, right? And this, most people agree, is sort of the founding moment in immunology. This was the moment when someone realized, you know what, this isn't all just good luck. There are immune systems that protect us. And actually, for that discovery, jointly with Paul Ehrlich, Mechnikov was awarded the Nobel Prize a few years later. Since that time, in 1882, we have come to realize the immune system is just extraordinarily complex. In fact, it essentially rivals the most complex organ we know, which is the brain, in terms of its complexity. And to kind of imagine that, so as a rough approximation, for those of you who are adults in the audience, your blood system currently is about 1% white blood cells, so immune-focused cells, and that's in the order of about 10 billion white blood cells. You've also got lots and lots of white blood cells that we can't count, distributed in your deep tissues. They're in your muscles and in your organs and in your brain, doing all sorts of jobs there. And then if you get an infection, the number of white blood cells goes up. And actually, very, very swiftly, it comes close to the 80 billion neurons that you have in your brain. So in terms of the absolute number of cells you have, your immune system is not that far behind your brain. But also, of course, in the level of communication with each other, these cells also undergo this incredibly complex part of biology which controls our immune response, just as your neurons are talking to each other. And we, just like as with the brain, we still understand very little, despite the fact that over the last 140-odd years of immunology, we have had literally thousands and thousands of people working on this problem. And as a quick count, just a couple of days ago, I had a look on the internet, and if you look for academic papers with the word immunity or immunology in the title, there are in the order of 5 million papers out there. And just to give you a sense of scale, if you were to sort of read at the average adult's reading speed and start now and never break, not even to go to the toilet, you'd finish in about 120 years from now, so you better get started. So there's an awful lot of stuff about immunology, and yet still a massive amount of stuff that we just don't understand. And that is because the immune system is doing an incredibly complex job. And at a kind of first approximation, the job it is doing is protecting you against absolutely everything, right? Your immune system needs to protect you against viruses and fungi and bacteria and all the things that we don't know of that are bombarding our bodies all the time. And so it's acting like a kind of standing army. And just like a standing army, this is this famous painting of the Battle of Waterloo. And in this painting, what you see are all these different specialized troop types, right? You have people on horses and you have people on the ground, you have cannons in the back. You have all these different specialized armaments. And your immune system is just like that. You cannot protect yourself against all these threats if all your white blood cells are the same. So you have all these different white blood cells that are talking to each other and coordinating with each other in order to minimize the threats they get. And let me give you just an example of that. This is actually some work from my own group, which is a rarity in a Gresham lecture. So this is exploiting an advantage of zebrafish. So zebrafish, some of you might know if you have a tropical aquarium at home, are these little fish that you can get in your tropical aquarium. When they produce young, one of the big advantages, those young hatch, and for the first about 5 days, zebrafish young are transparent, which is a significant bonus if you're a biologist and want to study stuff going on. The other real bonus about zebrafish is that you can anesthetize them incredibly well by putting stuff in their water. They fall asleep for very extended periods of time, and then if you change their water, they wake up again, swim off like nothing ever happened. So you can do some very cool experiments in zebrafish without really hurting the zebrafish. And here is an example of it. So this, to orient you, is the back of the head of a zebrafish. Okay, so it's lying kind of like this. And you're looking through the back of its transparent head. And in this experiment, what we have is a fish in which 2 different types of white blood cell are labeled. They're red and the green ones here. And this fish has a problem because it has a fungal infection, which are the blue cells you see here. So it has a fungus that's trying to grow inside its brain, which is a very bad idea indeed. And what you'll see, if we run this time-lapse movie, is the level of coordination as those different white blood cells recognize the pathogen, start to aggregate around it, and what you can't see at this level, but you can kind of interpret, is the conversation that's going on between these different cell types as they wrestle with this invading pathogen and try to clear it, all in the matter of a few hours. You can see the timeline here, we're about 8 hours now. So even in this incredibly simplified model of a larval zebrafish looking at only 2 of their white blood cell types, you can see a really elaborate conversation going on, a complex piece of biology that is dealing with this infection. And it's that really, I think, that I'm particularly interested in, and I think where there is a huge amount to be discovered and potentially to be harnessed for human good in understanding this complicated arrangement. So what actually happens when you have an infection, or indeed when you prick yourself with that kind of thorn that Mechnikov used back in his early experiments? So what happens, right, is you have put something that is not supposed to be there inside your body. And that could be bacteria, it could be a fungus, it could be a virus, it could be actually just a piece of wood from a blackthorn or something, but it's something you don't want there. And just as with any kind of conflict situation in real life, what you want is a very rapid response. If you think about that, if it's a bacteria, for example, so a single bacteria, under good conditions, might replicate itself about every 20 to 30 minutes. So if I put a single bacteria on the end of that thorn and inject it into somebody and do nothing about it, by tomorrow morning, that single bacteria has become 30,000 bacteria, and that organism is in serious trouble. It's no good being slow. If you have an immune response, you need to be very, very rapid. So you need a first response that recognizes and responds very rapidly to infection. And you can see that here, those of you who were at the last lecture, you've seen this movie before, and you can doze off for 30 seconds. This, again, is a zebrafish. In this case, we're looking at the tail, though. And here in the tail, this zebrafish has got a tiny cut. It's the kind of scratch that you might have from a paper cut or a grain of sand or something like that down here. The scratch is not sterile, right? It's introduced things into the tissue of the zebrafish that shouldn't be there, and the zebrafish recognizes that, and its immune system responds. And what you see are single cells diving out of this nearby blood vessel and racing their way towards this wound site to recognize and eventually kill the bacteria that should not be in there. This response is really critical. In this case, what will happen, in fact, if you ran the movie for another few hours, what you'd see is the wound heals and the fish carries on and swims off. If you don't do that fast enough, within an hour or two, the bacteria in this wound will have started to grow, take over, and you'd have a really serious infection. So the speed and the accuracy of that response is really critical. And I have to say, so we're doing these experiments in zebrafish because they're feasible. This is exactly the same process that happens when you cut your finger on a paper cut or a grain of sand. So what are those first responder cells doing? What they're doing, just like any kind of first response unit in an army, for example, is they are trying to neutralize that threat very rapidly. And the way they're neutralizing it is very simple at one level, which is they eat it, okay? And so this is what they look like. These are these kind of first responder cells. These ones are called macrophages. And in this experiment, we're giving them little plastic beads. You see one here. It knows that's not supposed to be there. It will reach out in a second, grab that bead, and scoff it. And you have to imagine this happening now within a tissue. So within one of those scratches, you've got lots of bacteria. These cells are racing their way in. They are recognizing the foreign object and eating it. And that's a pretty effective way to retain, to constrain that initial infection. Because if you can eat all these bacteria before, for example, they enter the blood system, you will reduce the rate at which the spread of the disease can happen. That raises a big question. The big question is, how on earth do they know, right? How does that first responder cell that has happily spent its entire life zipping around in a blood system realize that all of a sudden there is a bacteria somewhere else that needs to go and deal with it? And actually, the same person who did those early experiments in starfish larvae, Mechnikov, also figured this out. Here he is here, together with another famous Russian genius. This is Leo Tolstoy, him of War and Peace fame. I'm really curious in this photo. What were they talking about? What was the guy who's a sort of immunologist and the guy who wrote War and Peace talk about over tea? But anyway, so Mechnikov, having not kind of sat back on his laurels and kind of discovered immunology, that'll do, asked himself the question: these cells that have responded in the starfish larvae, how did they know that's where I'd pricked the thorn into? And he correctly surmised there must be something foreign, some kind of foreign molecule that attracts these cells, something that stimulates them and says, I'm not supposed to be here, come and deal with me. Something that we now know as to be a chemoattractant. Okay? And what Mechnikov speculated, and he was correct, is that there must be things that particularly microbes have, molecules that microbes have, that are foreign and therefore easily identified as something that you can use to home in on an infection and deal with it. And so, for example, this is a typical bacteria here, and many bacteria have these long flagellae, which are things that help them swim around. We, mammals, don't make the protein that produces these. So this is a very powerful chemoattractant. If you put a little bit of a bacteria tail in somewhere, your white blood cells know there's a problem, they home in on it very rapidly. And this really is what is driving that early response. So you've got something into a wound, something's not supposed to be there, your white blood cells are programmed to home in on and be attracted to any of these foreign molecules. And they do this incredibly well. And this is perhaps one of the most famous movies in all of immunology. And what I really like about this is that this was made in the 1950s, which is even older than I am. And it was made on 16mm film. So if you see these films where people go like that, imagine that. So David Rogers, who made this film, had this thing going on a microscope and made this film in the 50s. And it is still used today as the prime example of this process in action. And what you're going to see in this rather lovely film is a white blood cell here, this big chap in the middle, which is called a neutrophil. It's one of those cells that responds very, very rapidly to infection. It's the same cell, actually, that you saw zipping its way around the zebrafish tail a minute ago. This is a blood smear from somebody, and so these round cells here are red blood cells. And what you can see in this blood smear is you have a neutrophil, you have some red blood cells, and you have this chap who's not supposed to be here. This is a bacterium, okay? And this neutrophil knows that bacteria is not supposed to be there, and look how well it does when it knows that. So here's your bacteria, here's your neutrophil, here's your red blood cell, and off he goes with just an amazing amount of dogged determination. I think there should be a soundtrack to this, really. And turning incredibly rapidly over speed, and eventually, this is not the movie in which the underdog wins, I'm afraid. Hold tight, don't put your money on the bacteria, because he's about to meet his maker any second now, I think. But the accuracy with which this single cell can find and eventually engulf that bacteria is incredibly precise. Bear in mind, right, so this cell here, this neutrophil, is about 10 or 20 microns, so you'd get something like 20 of those cells end to end across a single human hair. So the scale of resolution is incredibly accurate. They can hunt and engulf those organisms that aren't supposed to be there. On its own, that is already quite useful in the sense that it prevents the infection from spreading. But of course, it's not very good if you keep the bacteria alive, because eventually they will get out and they will cause problems. So what you have to do when you've done that, when you've hunted them down and you've engulfed them, is you have to kill them. And the way they do that is essentially by using chemical warfare. Inside these white blood cells, these neutrophils, are little packages that are full of, it's like the kind of contents, under your kitchen sink. You have one of these kitchen sinks where underneath it you have all these sprays that say, kills 99.9999999% of everything that you've ever seen. It's like that. Inside these cells are little tiny packages that have all sorts of things in them. They have reactive oxygen species, so those are the things that damage DNA and cause mutations. They produce hypochlorite, bleach, within these little things. They have poisonous proteins that degrade cell walls. They're kind of like the ultimate sort of killing device inside these little packages. And when they recognize and engulf something foreign, they mobilize these toxic granules to destroy the thing they've eaten. And you can see that in this rather wonderful movie that's from Max Gutierrez, just across the road at the Francis Crick Institute. So now we're looking at the top of one of those white blood cells using some kind of fancy imaging in which these toxins are labeled. So the brighter the colors, the more toxins. And what you can see in this resting cell is that it has these little packages that are kind of laden with this sort of dangerous weaponry, but they're just sat there doing not very much. What's going to happen to this cell, though, is it's going to be exposed to a foreign particle, in this case a small bead, several small beads. And what you'll see, you can't see the beads because they're not labeled, but as they land, you'll see very rapidly that they land, and very, very swiftly, they are surrounded by these things, and they pour the contents, these toxic contents, into that bead to try and kill it. And this is the process that is protecting you all the time. So on your way in tonight, especially those of you on public transport, you will have picked up all sorts of horrible stuff from the people next to you in public transport. Even those of you who came in with each other, you've picked up stuff from each other, not good, and you need to kill it. And so what you're doing, in fact you've done it by now because we're already 15 minutes in, is your cells will have recognized all the foreign rubbish that you've breathed in or scratched into your skin from your journey in. They will have eaten it, and they will be doing this as we speak. They will be pouring toxins into those bacteria and fungi and destroying them very, very effectively. And this is absolutely essential. We know that because if you completely lack these cells that do this, you're in serious trouble. So if you have no neutrophils, which is this type of cell here, you're what's called neutropenic, and you're at extremely high risk of all sorts of infections. And actually, this is one of the cell types that is particularly damaged by things like chemotherapy for cancer. And it's one of the reasons why people who are undergoing these kind of treatments have to be very, very carefully protected from a lot of the infectious threats out there. And there are some other cells, macrophages, the first ones I showed you, that as far as we know, you actually can't even live without. We have never found anyone who doesn't have macrophages. So presumably if you lose your macrophages, it's just not compatible with life. And so these are protecting you to such an important extent that they are kind of critical parts of your armament. But even if you have them, their function, like all cells, varies a bit more or a bit less. And actually, this is one of the areas of immunity. People often ask me, well, what can I do to boost my immune system? And boosting your immune system is not really a thing, but maintaining it at a good level is something quite important. And these are a cell that respond quite rapidly to things that you do in terms of your kind of lifestyle. And so one of the things, for example, that these cells really need is vitamin D, which is this molecule here. So vitamin D, as you may know, you get in your diet to some extent, particularly from animal products, not so much from plants. But we also make vitamin D in our skin. So if you're exposed to ultraviolet light, sunlight, your skin will produce vitamin D for you. And in a sunny day, when you're out and about, even in London, you will generate enough vitamin D to be fine. But this time of year, when most of us don't see very much sunshine at all, particularly if you have the kind of job where you're not out during daylight hours, or indeed if you have dark skin, you produce less vitamin D, and you can very easily become vitamin D deprived. And actually, if you have low levels of vitamin D, one of the first functions that starts to be impaired is that killing function, that ability to destroy bacteria or fungi that you've eaten, which is one of the reasons that low vitamin D levels are correlated with increased risk of infection. And it's one of the reasons why we encourage everyone in the UK to take some level of vitamin D supplementation, particularly in the winter months, to keep your immune system on tip-top order. Okay, so what we've talked about so far is all about that kind of first response. And actually, everything I've talked about is happening really in the first few minutes after that initial wound or that initial infection. But it's only really half the battle. If you only had an innate immune system, you wouldn't do very well long term. Because what you need, like any decent army, is not to just deal with the initial incursion and then you all go home. You need to think about coordinating a much more elaborate long-term response. So if you've responded to that initial infection, imagine you're now in your kind of military analogy, you've dealt with your interlopers here. Most of the time, you don't just go home, right? And the same thing is true, I guess, for many of us in terms of our domestic lives. If you've gone home, I don't know, you go home after this lecture, you open your front door and there's some, bloke in your lounge pinching your TV. Even if you shout at them and they run away, I guess, hopefully, most of you won't just say, oh, well, that's good, that's enough now, I'll go back to bed. You might do some other things, right? You might, for example, go and check with the neighbours and see if they're okay. You might raise the alarm, phone the police, do all those kind of things. Your immune system does the same thing. So having initially dealt with your infection, it then realizes that this may not be the end of the story, and it needs to trigger a broader response. And that response is what we recognize as inflammation. So typically, right, when you sort of injure yourself or something, there's quite an obvious response to that. Maybe I'll pick on somebody in the audience. I'll pick on this gentleman here since he's right here. So imagine you've scratched yourself on that thorn. What's your symptom?
Blood and
Inflammation. And what does inflammation look like? Swelling. And? Hot, red, grim? Yeah, exactly. Very good. So right, inflammation is that response, and we all know this, right? You prick your finger, you cut your thumb, after you finish swearing, what you see is your finger gets red, it gets swollen, it gets hot, and that's not because of the infection, right? That's not bad stuff happening. That's actually good stuff. That is your immune system kicking in, realizing it needs to do a bunch of things. So when it's doing this, what it's actually doing is summoning in additional help. So it is triggering, for example, your blood vessels, your nearby blood vessels, to widen and to loosen, so that it allows more white blood cells to come more readily out of surrounding blood vessels. It's sending signals much more widely. It might do things like elevate your blood pressure. It might elevate your core temperature. You might get a fever. All of these things are designed to make sure that whatever the infection was, it can't spread. And there's a really beautiful picture here, I think, of this actually happening in kind of real time, if you like. So here, right at the middle of this picture, is a single green cell. That is one of these early responder white blood cells, and it has found something that's not supposed to be there, bacteria, for example. When it has found that, it's eaten it and it's dealt with it, but it has also started to produce a protein and secrete a protein that warns other cells, I've seen something bad, you need to be ready. And what you can see in these surrounding cells is when they detect that protein, they start to produce a pink color. And what you see quite nicely, I think, is the cells nearest to it are going very pink, and it gets less and less over time. And actually, if you look close down here, there's another little green one, and you can see a little flurry of pink cells here. So this is kind of your immune system sounding the alarm and encouraging other cells to come and help do with it. And it's that process that is driving this. So in this movie, what's happened is these early cells that have responded are sending out signals and calling in their friends from the nearby blood vessel. And if you could look at this at the level of the whole zebrafish, what you'd see was it's slightly swollen because there's more cells in that space. It's hot because there's a lot of cellular activity going on and all the things we associate with inflammation. And that is great. Most of the time that is great. That's really important. It helps keep you alive, basically. But sometimes it goes pretty badly wrong. Sometimes it's irritatingly wrong. Sometimes it is life-threateningly wrong. So this is one of the times when it goes wrong. For example, this is rheumatoid arthritis, which I suspect some of you in the audience might be experiencing. This typical kind of swollen knuckle here. This is inflammation, right? But what is happening here is this is not helpful inflammation. This is unhelpful inflammation. Your immune system, in this case, has overreacted to your own protein. It is recognizing a protein in your joints as foreign, and it's doing all the things we just talked about, but against your own joints and not against an invader. So it's also encouraging other white blood cells to come in, it's changing the blood vessel patterns, it's changing swelling, and that's why you get these kind of hot, painful knuckles, because you're triggering the same process, but now unhelpfully against your own joints and not against a foreign threat. This is a more serious and quite common partially genetic disorder. This is lupus, and lupus is still a very poorly understood autoimmune condition in which inflammation is targeted at your own tissues, sometimes in a life-threatening manner, and it has all sorts of old manifestations, but one of the most classical, which you can just about see here, is this so-called butterfly rash on the face. Again, in this butterfly rash, what is happening is exactly the same process. there is an inflammatory response going on. It's calling in white blood cells, it's loosening blood vessels around there, and you're getting this rash. And in some cases, lupus can be very serious because you can also have this happening, for example, in your kidneys or your liver and cause organ failure. And so understanding this balance of helpful inflammation versus unhelpful inflammation is really kind of one of the big challenges, I think, of modern medicine. And of course, many of us experience the whole immune response, inappropriate immune response, very commonly, particularly in spring. So for some of us, you'll be looking at this picture going, oh, what a wonderful holiday scene. And for some others in the audience, you'll be thinking, this is my idea of a nightmare because I take one step in this field and I end up like this poor lady here. Because, of course, many, many people have hay fever. Hay fever is exactly the same process. It is an inflammatory response to a foreign protein, except in this case, it's a theoretically harmless protein. It's a pollen protein, but your immune system doesn't realize that. It thinks that pollen protein is just as dangerous as a bacteria or a fungus, and it is launching the same level of response. And actually, in this particular case, because what you're doing is inhaling the pathogen, the key white blood cell that is responsible for this response is called a mast cell. And what mast cells do is they sit primed, particularly on your airways, looking for foreign things, and they respond quite accurately to foreign threats like bacteria. But in this case, if you have hay fever, you're responding inaccurately to pollen as a foreign threat. And what you do when you detect it is you release a molecule, histamine, which is a kind of serious alarm signal to the immune system. So when it releases histamine, that is shouting at your immune system, you're in real trouble here, get ready. And actually, there's a beautiful movie you can see here. So on this movie here, we're looking at the base of a mast cell, and you can't see it, but we've looked for histamine. And what you'll see is when you add pollen to this particular thing, hopefully if the movie plays, is that the mast cell responds by releasing histamine in little tiny puffs, and actually it looks like some kind of fireworks eventually. It looks really cool, unless it happens to be your mast cells and your pollen allergy that's driving it. So this balance between early response, inflammation, and then broader immunity is quite key. And that, for many organisms, is as far as it goes. So for most invertebrates, for example, like mollusks or insects, their entire immunity relies on this innate response that we've just been talking about. And that's partly because if you're a short-lived insect, you don't really need to worry too much about repeated infections, right? If you're a fly that might only live for a month, you don't really need to worry so much about whether you're going to see the same infection next year, because next year is not going to exist for you. But if you're a long-lived vertebrate like us, having an immune response which only deals with things over a few days is not very good, because we live long enough to encounter the same pathogen again. And so what vertebrates have evolved is this process that layers on top of that early response called adaptive immunity, which gives us a long-term, kind of lasting memory and an incredibly precise response to pathogens after they have caused an initial infection. And really, it's the difference between this kind of slightly crazy, disheveled innate immune response, which is effective, but it's kind of a bit discoordinated, versus this really precision, slower, but extremely powerful adaptive response that drives long-term resistance. And actually, in a later lecture in this series, we'll talk a little bit about how this has evolved and where it's come from. But one of the most interesting things, I think, is how these two systems are interplaying between each other. So that early response, even a really, really mild infection like a scratch or something or a cold, will be dealt with very rapidly, but will still engage this wider adaptive immune response. And the key cell in our immune systems that bridges those two between the innate immune system and this long-term adaptive is this guy here, the dendritic cell. I really like these, not least because they're very funky. So this is a dendritic cell. And dendritic cells are called that because dendritic is a word meaning kind of branch-like, lots of branches. And you can see all these kind of things out the side. And what dendritic cells do is they sit in your tissues and they literally just eat for a living. They're eating everything all the time. And you can see this guy here eating everything. And in this case, a passing yeast cell gets captured and scoffed by this dendritic cell. And they're doing that not really to deal with these infections in the same way as the cells we just talked about. They're doing it to sample what is there. So you can think of these things as sort of security guards in your tissues. They are testing the whole time and seeing what's there. And if everything they're eating is other human proteins, then they're not bothered, they're fine. As long as they're just sampling your own tissues, no worry at all. But if they detect something that is not a human protein, like a foreign yeast here, then they are triggered to sound a much broader alarm. And I think these things are basically like kind of Cookie Monster up here on the side. They're spending their whole time scoffing and scoffing and scoffing. And just like Cookie Monster, actually, if you zoom in on Cookie Monster, what you see is that having scoffed his cookies, he then displays the stuff he has caught to the rest of the immune system. And that is what these cells do. They're eating the whole time, and then they're displaying the cookies they've eaten to the rest of the immune system. And they're doing that in order to engage other specialized white blood cells. And so that's the interaction you see here. One of these dendritic cells has eaten something foreign. It knows it's foreign, and it is presenting fragments of that thing it has eaten to, in this case, T cells, other sorts of white blood cell. And it's doing that a bit like one of those photos you see sometimes in a supermarket or whatever. Have you seen this person? They're a known shoplifter. It's essentially telling your immune system, I've seen this, it's not good. If you see it, you need to deal with it very, very fast. It's an alert system. And it is this process that underpins essentially all of kind of our modern approach to immunotherapy, to vaccination, to long-term health benefits. Because if you can instruct the immune system to be ready and primed to deal with a threat, then you can head off infections before they ever get a foothold. So what actually happens with one of those cells? So one of those dendritic cells, they're scattered all through your tissues, particularly in places where you're likely to encounter some foreign molecules. So for example, in your lungs, on your skin, your mucous membranes in your mouth. They're sitting there the whole time sampling. When they eat something that they recognize as foreign, they decide to get up and move, and they start migrating to the nearest what's called a lymph node, a draining lymph node. And so you can test this at home. In fact, you don't have to test it. You will undoubtedly experience it at home. If you get some kind of respiratory infection, so a sore throat, a cough, a cold, often one of the first things that happens right is you feel these lumps in your neck swell up. Yes, please somebody nod. And what you're experiencing here, as well as being very irritating, is exactly this process. Dendritic cells in your mouth and your throat have detected a foreign particle, in this case usually a virus. They have eaten it, they have got up and they've started to migrate to their nearest lymph node. And from your mouth, the nearest lymph nodes are the ones in your neck. If you cut yourself on your leg, they'll go to the ones in your groin. If you cut yourself on your arm, they go to the armpit. And sometimes you can feel that, right? If you have a cut on your arm, you might feel a swollen lymph node in your armpit. The dendritic cells have done that because at the lymph node, it's like the kind of bus station for the immune system. Or lots and lots of different white blood cells are passing through a lymph node all the time. So if you're an activated dendritic cell, which are the green cells here, you can sit in the lymph node and you'll see lots of white blood cells come past and you can wave your flag at them and say, I've just eaten this virus, you need to go and deal with this. And that's what you can see here. So the yellow cells are T cells zipping through the lymph node. And what you occasionally see, there's one over there, is these green dendritic cells sort of stick tight onto a T cell for a few minutes. And in that conversation, they are telling the T cell, I've seen this and you need to go and deal with it. What you have in your body are literally millions and millions and millions of different T cells, all primed to recognize a different sort of molecule. So in practice, what's happening in your lymph node is that thousands of T cells are looking at the dendritic cell saying, nope, never seen that guy, nope, never seen that guy, nope, no. But they're looking for the one T cell that says, yeah, I know that, I'm targeted to deal with it. So they are priming the T cell that has a specific response to that virus, for example, to say, this is your moment. You've spent your whole life waiting for this virus, and I've just seen it. Now it's your job to go and get it. And what will happen after that interaction is that the T cell that is specialized for that particular virus will go off and replicate very, very fast into lots and lots of copies and now go and start to deal with that particular infection. So it's an incredibly precise way of dealing with a very precise pathogen. So you're not alerting the whole immune system, you're just alerting the bit that you need at that one moment. And you're alerting it because it's the adaptive immune system, it's these T cells that enable you to do two really, really important things. One is it will trigger the production of antibodies against the thing you've just had, right? Against that virus in this case. And it does that because having activated one of those T cells and so this is your moment, this is the virus you've seen, the T cell will go off and talk to another cell called the B cell, which look very similar, that matches and say, okay, produce an antibody for me that matches this particular virus. And we'll talk more about that in a later lecture when we talk about vaccination. And so the first sort of response you see of this adaptive thing is when you start generating antibodies against that particular virus. And in fact, if you're sick for a long time and go to a doctor, they might say, I'm going to check you for antibodies against this thing. And actually, that's the basis of, for example, whether we know people have had influenza or COVID, is you can look for antibodies in their blood. And if you see the antibody, you know that this process has happened and that person has been primed to produce antibodies against it. Antibodies on their own are really effective. They will do all sorts of stuff that helps limit the spread of the pathogen. But actually what you really need is the cell type that will eliminate your pathogen rather than just label it up. And for that, the final white blood cell of our lecture is introduced, which is the killer T cell or cytotoxic T cell. And this is a type of white blood cell whose entire function is to destroy other infected cells. So if you think about an infection, when you get an infection, many infections are extracellular, right? So bacteria or fungi tend to grow outside of your tissues. But one of the biggest threats we have are viruses, and viruses, by definition, can only live inside your cells. They get into your cells, so COVID-19 is a great example. It enters your cell, and it can only replicate itself inside your cell. Good for the virus, less good for your immune system. It's very difficult to see a virus if it's hiding inside your own cell because the surface looks like a normal human cell. And so these killer T cells have evolved deliberately to target those hidden intracellular viruses. And they do that in an incredibly effective way. What they do is they look at the surface of all your cells for the presence of any foreign proteins, in this case, a viral protein. And if they find that protein on the surface of your cell, they will kill and destroy the cell that it finds it in. So it's a bit dramatic, sorry cell, you're going to die too, but it's a great way of stopping the infection spreading because you take out your own cell, but you take out the virus that's inside it too. And they do that in the most elegant fashion ever. So this is a fabulous movie from Gillian Griffiths. So here you have a T cell, and here you have, in this case, a kind of fairly standard human cell that is infected. Okay? And what you'll see is just how carefully that T cell looks at the surface. It's rumbling all the way across that other infected cell, looking for the presence of a foreign protein. And if it finds the foreign protein, so if it doesn't find it, what will happen is it will disengage and carry on, leaving the blue cell to be and doing its own thing. But if it does find it, and it says, okay, now I realize that this blue cell is infected, it triggers an incredibly effective killing program. Inside that T cell, which is now green in this image, are all these little red granules. And these are very similar in some ways to the granules I showed you at the beginning. They are toxic granules that kill other cells. They've got different toxins in them, but they are kind of the kind of hand grenades of the T cell universe. So the T cells are laden with these granules. They're looking at these other human cells that happen to be infected, the blue cells here. When they find an infected cell, what you'll see is they move these little killer granules very close, and then you'll see we pause the movie in a second. So it gets close, moves its red granules in, and then we'll pause the movie, turn it around, and what you'll see is the granules are sitting just above the surface, and then they'll be released. Plop, plop, plop. Into that target cell. And if you're that target cell, this is kind of the last thing you'll ever see, because this is the moment at which your T cell is delivering that killer blow. And it does it in pretty dramatic style. So you can see here now, in this case, T cell coming in, infected cell, delivers those red granules, and within a couple of minutes, bingo, it's all over for your infected cell, and of course, the virus that's inside it. And it is this process that is protecting you from an enormous number of foreign pathogens. So many, many viruses, also some intracellular bacteria. But it's also, and that's a relatively recent discovery, it's also happening all the time to control cancer in your own bodies. So all of us, all the time, essentially, are at risk of cancer, right? Our cells are dividing, mutations are happening, DNA mutations are happening. And actually, we now know that over the course of our lives, all of us will generate these kind of micro-cancers. But actually, there is a hugely important role for your immune system in stopping those micro-cancers becoming real cancers. Because some of these T cells spend their lives looking not for foreign proteins from viruses, but for foreign proteins created during this mutation process that are a hallmark of cancer. And when they see them, they destroy these cancer cells and prevent the cancer from growing before you even know about it. And it's this process that we are now trying to harness through immunotherapies for cancer. So the idea being that at the moment, what we typically try to do is to stimulate the immune system to help remove a cancer in conjunction, for example, with chemotherapies. But where people are really just starting to sort of scratch the surface of now is the ability to perhaps immunize people against cancers. And there's quite some very promising work where if you have a tumor, people can identify in that cancer a protein that's present in the cancer but not in the rest of your body. And you can kind of vaccinate the immune system and encourage it to then sweep through the body and remove not only that primary cancer, but any invisible metastatic cancers that are elsewhere in your tissues that you might not be able to see if you're a doctor, but if you're a T cell, you can still find them and destroy them in this way. And that process of immunotherapy, I think, personally, is one of the places where we have a really decent chance, actually, of ultimately turning the tables on cancer and many other diseases, actually, and really using the power of this precise T-cell targeting to help cure ourselves of disease. And on that relatively optimistic note, let me finish there, and I'm very happy to take any questions. Thank you very much.